Hello, my name is Dr. Bradley Alicea, and I'm going to be presenting today on phylogenetically-mixed architectures, the development and evolution of agentive complexity. I'm from the Orthogonal Research and Education Lab, and this is being presented as a poster at Artificial Life 2026. So when we think of something as phylogenetically mixed, it's this concept of phylogenetic mixing. And phylogenetic mixing can be seen as an in silico hybridogenesis. What does that mean? That means that we use phylogeny to determine sort of endpoints of evolution or lineages of evolution. So here we have worms and flies and mice and primates, and they all have their own, they all come from different lineages. Sometimes they have shared characteristics, sometimes they have unique characteristics. And when we take those endpoints, they typically have different nervous systems that are specialized for different things. And typically in nature, you don't see a lot of hybridization. You sometimes see hybridization, but it's limited to very closely related taxa. What we do here is we evolve agents, and we evolve agent architectures that are specialized for different things. And then we put them together after they've evolved, and we end up with this composite neural architecture. So different nervous system circuits or different agent phenotypes, different agent embodiments equals a composite neural architecture or a composite agent architecture. So we can place these inside of a generic body or a hybrid body with features of the circuit in question. So our first objective is to evolve from a common ancestor. So we have some sort of common ancestor. Maybe it's sort of a common plan. We're going to see in this paper that we use abstract shapes and we use Bradenberg vehicles as our two test cases. And we evolve our system from a common ancestor. We have a simple motif that we start with, and then we generate, through a generative process, variation. And then we select that variation. We end up with these unique lineages, and then we put them back together as, in the case of what we're doing here, a hypergraph, which then defines the architecture. And the architecture then can be as big as we would like. We can scale that architecture up quite a bit. The key is generating the variation and selecting upon that variation that give it this sort of ability to kind of be placed together in this bricolage. Our second objective then is to stitch phylogenetically distant motifs together. So if you see in this example, it's not a great example because you wouldn't do this necessarily in a natural system, but if you see our phylogeny on the left, we have our worm, our fly, our mouse, and our primate, and they have different evolutionary histories. The mice and primates share closer ancestors than the fly and the worm. But what we're doing here is we're putting together a neural circuit where we combine elements of the worm architecture with the primate architecture with the mouse architecture. And they're all in the same structure, or all in the same agent. So we're stitching phylogenetically distant motifs together into a functional whole. Now, there's prior work on the top-down generation of embodied agent forms. So one of the things we're doing here is we're proposing a pipeline for generating embodied agent phenotypes or embodied agent architectures. And so in the field of morphological computation, there have been a few attempts at building a pipeline that allows you to build these optimal architectures for robots and other types of embodied agents. So one of the first attempts was Cheney in 2017. This was from a dissertation. They proposed a design pipeline that establishes an optimization mechanism for specialized and optimized complex morphologies. So again, not doing this sort of phylogenetic mixing, but basically optimizing different complex morphologies. Then D'Angelo et al. In 2026, this is a paper where they offer this design pipeline for embodied agents that allows us to build that allows us to build architectures that we can benchmark against one another. So it's becomes an important component of benchmarking agent performance. Now, if we compare that with phylogenetic mixing or this in silico hybridogenesis, our approach provides a means to develop novel and consensus forms, while also assembling more complex morphologies from disparate components. So one of the things we're aiming for here is something that we presented at Embodied Intelligence 2024. So I talked about agent embryogenesis. And what agent embryogenesis is it's developing embodied agents. So we see here we're developing, if we look at the top sequence on this slide, we're developing a Bratenberg vehicle. And we're doing that from a single sphere, which is kind of like a fertilized egg. And this developmental analogy continues where we have this fertilized egg, quote unquote, and then we move on to this, what we call trypoblastic organization, where we have 3 germ layers of tissue. In this case, it's just material type. And then we have movement or inversion of those different layers, where we end up with sensors, effectors, agent body, and agent nervous system. And each of the different layers contributes to those different structures. And then we get refinement, and then we get shape transformation into the vehicle that we see on the right-hand side of this sequence. So this is something I talked about in that talk as the trypoblastic model of embodiment. And so in this case, agent embryogenesis, which we defined in that sequence, proceeds as a series of morphogenetic movements or acquisitions. So in a system where we're building these agents, we start with the trypoblastic model. We allow for development to sort of take shape. So there are a lot of inversion events, a lot of transformations of the body. And these are all acquired in a series of steps. So sometimes those steps change for different agents. We can intervene and change those developmental steps. We can change the timing, and we can get different morphologies. And we can control this with the genetic encoding. I'm not going to get too much into the details of this, but basically we have this system where we have the series of acquisitions of morphological traits. We also have these acquisitions of behavioral traits. So the behavioral traits tied to these morphological traits. The morphological traits need to be there for the behaviors to become manifest. And then the whole thing is this developmental process of building these vehicles. Now, if we dive deeper into this trypoblastic analogy from developmental biology, we find that we can actually do some really interesting things with developing agents. So on the left-hand side of this phylogeny, we have a trypoblastic precursor, which results in certain types of phenotypes. In this case, we have these roller phenotypes that have wheels, so the vehicle rolls, and that comes from sort of a common precursor of this trypoblastic agent. So we have the 3 layers. The 3 layers are the sensors, effectors, the agent nervous system, the agent outer shell, and then that allows us to form these shapes of these rollers. We also have a dipoblastic form. In dipoblastic embryos, you only have 2 layers. And those 2 layers, dipoblastic embryos are the more basal condition in the tree of life. And so you see these in marine invertebrates, for example. Dipoblastic precursors give you a limited sort of range of things that you can differentiate towards. So here we have an inviable phenotype in the swimmer category, and then we have the swimmer that develops from this interaction between the 2 layers. So we get this swimmer with a tail. And so we can do a lot of things here, but what we need is some sort of computational generative process. So this is a generative process that we've mocked up sort of in a diagram. But if you had a computational version of a generative process, something that would just generate really novel phenotypes, or sometimes really average phenotypes, and lock on to certain combinations of things, and then put them in a situation where they're selected for, and then we get agent designs that way. That's what we need to sort of continue this work. And so this is what we're doing in this paper. So what we have here is we have a pipeline where we have generator of variation. We have something that sort of discriminates between good and bad things, or maybe viable and inviable things in a latent space. And then we have this genetic algorithm with a fitness function that allows us to select the optimal forms. And so if you think about the last example, we can use a generative process to generate a lot of forms, a lot of potential forms. We can then use our discriminator to find the viable from the inviable forms. We saw one inviable form in that phylogeny, but we have other viable forms that can then be passed on to our genetic algorithm, which has some sort of fitness criterion that selects the things that we desire for our final sort of most fit phenotypes. And then, which I'm not showing in this pipeline, we have this last step where we have phylogenetic mixing, which we assemble things in. So if we revisit this pipeline, we have our first step is characterized by a reservoir network, but sometimes alternately we can use a diffusion process. And we try both of those in this pipeline. So depending on kind of the properties of your input data, you can either use a reservoir network or a diffusion process. And in the accompanying paper, we talk about the differences between reservoir networks and diffusion processes and what they're good for. So sometimes if you have unstructured input data or you have a lot of variation, the diffusion process is better than the reservoir network and vice versa. So that's why we have these two methods that are there. But in any case, they produce this function RT, which of course is the output. So we have the input, which is maybe something like the type of primitives that we would like to use, or it could be trained on data. Like in the case of our Bradenburg vehicles, we had to train the model on what Bradenburg vehicles look like and the different sort of primitive components that exist there. And so the output of that is this function RT. It's a time series. It goes into this GAN, which is a generative adversarial network. And a GAN has two components. It has a generator and a discriminator. So it takes this function RT, it generates variation in that, and sometimes it creates false positives, sometimes it creates the masked with noise. And depending on the parameters in the GAN, we'll talk about one of these parameters, beta, later on. And then the discriminator discriminates between the things that are viable and the things that are inviable, or basically evaluates false positives from true positives. And so for this GAN step, what we're doing is we're producing candidate latents and we're generating shapes. So this is the latent space. So our function RT goes into this latent space. We sample the latent space with the generator. We distinguish the things we want with the discriminator, and we pass that on to the next step with this function ZI. For our diffusion process, we have an additional step, which is the reverse diffusion process. And if you're familiar with diffusion processes, you know that there's a forward and a reverse diffusion process. So the forward diffusion process will take some set of, in this case, some set of shapes or primitives or whatever, and it'll add noise to those elements. So it basically obfuscates them. It trains the model to look for variation in those things by adding noise or adding some sort of corruption to the signal. And so that's the forward diffusion process. It encodes these, the training set as something that maybe looks different depending on the context. And then when you retrieve things from the diffusion model, you go through this reverse diffusion process, which is where you're retrieving things out of noise. And so the idea of a diffusion model is that adding that noise in the forward step improves retrieval in the reverse or the inverse step. And so that's why, when we use a diffusion process, we have to go through the forward process, the GAN, and then the reverse diffusion process. And that gives us this function S of I, which is basically this time series that we have that gives us this product of the same thing that we get with the reservoir network plus GAN. But of course, now we want to go and look at our selection criterion or our genetic algorithm. So for that, we have a genetic algorithm that we implement in Python. We have our fitness criterion. We have two fitness criteria, and we end up with this Z, which is what we get at the end. And then we can model that as a phylogeny. We can model that as a phylogenetic tree. We then can use that as as the grist for our hypergraph for our phylogenetically mixed architecture. So let's walk through this a little bit more. So the diffusion process produces a high dimensional state xt. It projects to a context vector ct. Sigma is the noise strength parameter. So a random signal is injected into the forward diffusion process, and this is if we're using a diffusion process. If we're using a reservoir network, we end up with our function by going through this reservoir network where we're sampling from the reservoir network. So a little bit different process there. Then we have our genetic algorithm where we sample from the context vector CT. This evolves samples and provides output Z, which we saw in the last example. If we look at our GAN model, this is a shape generator that receives this G, which is a combination of the context vector and Z. We have this generator which converts Z and CT into an 8-dimensional shape descriptor, and a discriminator which asks, is this a plausible shape? Does it resemble something in the training data? So the discriminator sort of confirms that what the generator is producing is something viable in this context. And so we're kind of switching the order here in this pipeline just for purposes of kind of understanding what this does. And then if we're using diffusion, we have this forward diffusion process that we talked about at the top. We also then have this reverse diffusion process that we can put at the end, which denoises the GAN output. So we can configure this in different orders. And there's feedbacks here, so when we run this pipeline, we can refine our results and go back up to the top and run it through. So there are a lot of ways we can configure this. So for this project, we actually use a technique called agentic coding. So why do we want to use agentic coding for this? Well, it's kind of hard to sort of figure out how to implement all these models together. And agentic coding is very good at generating code, and especially when you want to put it in the context of some problem. And agentic coding is basically being used here as an iterative research sketchpad. So this is where we have this sketchpad where we walk through a problem. We try to define the components of the problem. We ask the agent or a large language model, how do you generate a reservoir network for this problem, right? So you start off with maybe asking a few ways in which you could configure a reservoir network. So one of the things I started with was I wanted to generate a reservoir network that would give me a small connectome for C. elegans. And I was able to do that. And then I asked it if I could generate these shapes or generate a reservoir network that outputted different shapes, and then, kind of refine the model from there. And then I went to the next step and I said, okay, what if we add a GAN to that? What does the code look like? And if you keep doing this, it allows you to improve your pipeline, improve your model, and come up with a really sophisticated solution, at least in principle. Because as it turns out, you do need to do a lot of correction. You need to understand your problem before you can really kind of get deeply into this iterative process. But that's all part of the fun. And so this is sort of a newish type of research. We've been doing a lot of it in the lab. So this iterative research sketchpad provides a means to rapid prototype models. So like we do rapid prototyping in programming, in Agile. We also do rapid prototyping in sort of producing, um, if we think about the pipeline, we might generate the pipeline by hand and then refine it many times. So we're really not losing any time, or actually maybe not gaining any time either. But it gives us this way to do this where we can actually kind of go to a code base and kind of do experiments in a lot faster time than we otherwise would. And our development process is integrated with these refined prompts. So we keep refining the model by prompting the large language model and asking it questions about the different parameters it's generating, the different assumptions it's making about the model, and so forth. And from there we can build a more rigorous pipeline. We can also extract data sets from texts and use RAGs, which are specialized models, to make the model more specific. And in this case that I'm referring to is the Bratenberg vehicles example. So we'll see this Bratenberg vehicles data set, and we will basically train the model just on information about what Bratenberg vehicles look like. In this case, we're training the model on the book by Valentino Bratenberg, Vehicles. So it has all that information, and it's going to build a model based on that definition of vehicles. And so what agentic coding really is it's intelligence augmentation sensu Ashby. So W.R. Ashby proposed this model back in the 1950s. And he made the argument that if you work together with machines, you can, if you do it in the right way, you can really create this sort of superhuman ability. And I won't get into what that is, but basically our large language model is the amplifier, and X is the user, the human, and then G and U are the amplification process in this closed loop feedback. So we used Copilot for this work, and we'll talk later about how this work can be improved by maybe using other models. So this is what the dialogue looks like. On the top we have what the human is asking the large language model, and at the bottom we have what the machine generates in response. So these are prompts by the human, and the machine is responding, and this is a typical sort of interaction. So the human asks, what models are the most plausible for developing embodied phenotypes? The machine responds, the large language model generates code. So there's different steps in the analysis, handling of toolboxes in Python, things that might take a human a very long time to tweak or to really kind of get their head around. And so this is useful for implementing candidate models, especially when you have a lot of models that are coordinated in a pipeline. So then the human might ask, how does this fit into a pipeline? What do the details mean? I'll suggest what data to benchmark the models with. So this is all just kind of in preparation for building out this pipeline. And so we need to figure out the larger context of what the machine is generating. So the large language model refines the definition and technical details and helps to create benchmark data sets. The human then asks, I'll test the code and evaluate both the code base and the outputs. So the human actually, once the machine generates the code, the human has to verify that the code runs, and it generates what the human wants. And so this is a very important step, because you can't just throw the code into the pipeline and say we're done. This is a very hard step, because oftentimes you're asking it to do many things. It's generating graphs. You have to understand the graphs. You have to understand the output of the programs. And you have to understand sort of how it's treating different things inside the program. And so really it takes a lot. You have to really understand what the code is doing. And so in response to this sort of scrutiny, the large language model refines parts of the code and data set that do not make sense or do not execute. So you might ask the model, this didn't execute properly, and you might refine it and say what you actually want. And this happens a lot in agentic coding. And so then the human might ask again, I'll suggest ways to visualize the results using Matplotlib, and that gives the machine a way to sort of respond with graphs or with interactive graphs or something that's useful to human interpretation. So the first data set we use here is the shapes data set. This is a very simple set of primitives. We have squares, triangles, circles. And we use this because, we don't really want to start very complex. We want to see if we know what our shape ensemble looks like, we can know what the program is doing, how it's treating this in the latent space, how it's sort of discriminating good things from bad things, and how the genetic algorithm is locking onto the most fit thing based on our criteria. So our shapes data set is put into the pipeline. This shapes data set is sampled, and the pipeline generates ensembles of body parts. And you can see here on the left that this is our ensemble, and we will see examples of the ensemble in a latent space later. And then selection is used to explore the possibility space. So we use selection to sort of explore and refine the space to get the most fit output or the most fit ensemble given our criterion. And so we have this little test that we ran on this reservoir network. So in a reservoir network, you're looking at the level of noise, and that refines sort of the thing that comes out of the reservoir network. So this is where we have our sigma parameter, and we're doing a parameter sweep. We're starting with very low levels of noise and going up to very high levels of noise. You can see here that you have this, the latent space is changing with that parameter. As for our genetic algorithm, we implement this in Python, as I said. We have our crossover and mutation operators. And our crossover and mutation operators give us the opportunity to refine our ensemble. So we have oftentimes we can modify the mutation rate that changes what our ensemble looks like. We can also modify our crossover rate. And so we have this GAN output, which is ZI. This is fed to our fitness function, and then selection occurs based on one of two criterion. So the first criterion is smooth descent with modification. And this is where we don't expect anything radical to emerge from this process. We want to see basically an ensemble that is everything is very similar to everything else. And we want to basically sample our latent space in a way that does not disrupt what's in the ensemble. So we don't have these sudden jumps in the ensemble. So with smooth descent with modification, the criterion for selection is that we have the smooth transition between neighbors and latent space. So we select the smoothest transition between neighbors and latent space. So this is one sort of philosophy of sort of selecting things in our ensemble. We have this, what is basically respecting common ancestry very, very strongly. In the other case, we have creativity. So in creativity, and ZT is our function we're operating with. So ZI is the GAN output, ZT is the time series. And the creativity is where we penalize temporal dependence and novelty is rewarded. Uniqueness between neighbors and latent space is the goal. So we want to have uniqueness in our ensemble. We want to have things that are unique. Again, we don't want to have huge jumps in our latent space, but we also want to reward novelty where it exists. So we have less respect for common ancestry and maybe more respect for novel things that emerge in a lineage. And so this is just showing kind of this, what we're doing is, say, if we have the reservoir network and our GAN and our GA as part of a pipeline, a reservoir network is basically creating these motifs, these basal motifs that represent sort of common ancestor or some common ancestry. We're then generating evolutionary variation, and then we are selecting for specific types of variation to be represented in these terminal taxa of our phylogeny. So each terminal branch of our phylogeny contains different taxa, different endpoints of a lineage. And then what we'll do is we'll take these endpoints and combine them into a hypergraph later. But we do need to have this evolutionary variation generated, and we do need to have it in a way that is interpretable to sort of these phylogenetic relationships. So with our shapes data set, we start with a population of generated shapes, and then we select the most viable. We can do this with a reservoir network. We then have our GAN, and then we have our GA. This is an example of where we have our, on the left, what we've generated from our GAN, from a reservoir network output. So on the left, we have this latent space that is all the generated shapes that are generated by the GAN. So this is pre-selection. This is generated by the reservoir network. So we see that we have a good representation of the different shapes and different sizes, and it's pretty spread out across latent space. So no selection here necessarily, but we do have our viable shapes. And we do have a pretty uniform representation across latent space. Now on the right, we have something that's generated by the GAN plus the GA. So this is a latent space that shows when we have this evolutionary process that's active. So we have selection, and we have selection for specific shapes, where we end up with specific shapes and concentrated in a specific part of latent space. So we can see here we have all these triangles kind of clustered up towards the top middle section of the latent space. This is what our selection process does when we add the genetic algorithm. It restricts everything to that one space. Now when we have a fitness function, we basically, this is how we define it in the code with the symbolic variable z. And in this case, what we're doing here is we're creating an 8-dimensional vector. It's a one-hot transformation of shapes. We take the shape and we do a one-hot transformation. And then we have this, we're basically doing this from the previous time point in the GAN generator. So remember, the GAN output is this time series that we get to evaluate with our fitness function. And so there is this realism score that's generated from the previous time points GAN discriminator. Basically, the realism score is used to evaluate whether something is creative or something is hewing to common ancestry, or it's hewing to sort of the last time point. So the realism score allows us to implement these two different types of selection. The first way the realism score can help us here is that we have this center and size component. So the center and size component of the realism score judges the plausibility of generated shapes. Is it symmetrical and size scaled, or is it something else? Is it asymmetrical and not scaled to size? If not, then it's not viable. So that's one sort of component of viability. The next is smoothness, and we talked about that for both fitness functions, but smoothness is important because we want to make sure that large jumps in the latent space are not allowed. As we saw in the example where we had almost a nearly uniform latent space, we don't want to jump around latent space because we're sampling different things and we're not really getting a coherent sort of anything. So we end up with all of our taxa and our phylogeny being a mix of everything else, and it just kind of defeats the purpose of phylogenetic mixing. So our smoothness criterion is where large jumps in latent space are not allowed. No sudden transition, say from squares to circles. So we don't want to have this sudden transition. We do want to highlight, sometimes we do want to highlight creativity, but we don't want these sudden jumps that give us these sort of grab bag phenotypes. So this is an example of a GAN plus a genetic algorithm where we've evolved the steps over time. We have this, this is our latent space on the left with a population size of 100 and a beta value of 0.0. So in the GAN, there's something called a beta value, and the beta value is tuned to reflect what the generator and discriminator are going to do. So this is a beta of 0.0. We have these shapes kind of on the left-hand side of the latent space, and then this just shows the fitness of the genetic algorithm. So this is over 200 time steps for these rectangles, and this is just showing the fitness. So now the fitness, of course, is not optimizing. It's kind of bouncing up and down. So this is maybe one of the problems with this method, is we really have to work on tuning it to get exactly what we want. And so in this example, this is code for our GAN, and this is a function called the decode out tensor function, and this has a beta of 0.0, 200 time steps, and 100 or 200 generations for our GA. So we run this pipeline, and we have this diffusion process. Actually, this is where we have the diffusion process in the front, and then we have this fitness function. And so when we run our pipeline, we put all those things together, and the beta, of course, is going to determine what our kind of our stability of the model from the GAN to the GA. So when the beta is close to 0, shapes remain stable and the model retains temporal stability. When beta is 0.5, which is much higher than 0, shapes are less stable and the model loses memory. So basically what happens with beta, and we want to be able to tune this depending on our goals downstream, is that if we want to have stable shapes and we want to maintain temporal stability where the model can always go back to the last step to enforce this sort of regularity, we want a low beta. But sometimes we want a higher beta where we want to make the shapes less stable. We want to explore our latent space a bit more. But that comes with the trade-off of our model losing memory, and it becomes Markovian. So it loses its ability to go back to that last step, or rather the last step becomes less informative. It's just an example of how we can explore different parameters and how we can really take advantage of the parameters that are in the code to really kind of fine-tune and benchmark everything to the fullest extent of what's possible. Ask a question here: what if we change the fitness function to encourage novelty? Remember I said we have two different types of fitness functions. We have the creativity fitness function, and so we really want to try to encourage novelty here. So we have our fitness function here. We're using this center and size criterion, and we have this uniqueness score, which is basically allowing us to have a novelty first strategy in our selection criterion. So on the lower left hand side of the slide, we have this latent space where we have this GAN and GA output, where we have over time we have this latent space that's concentrated in the middle of the latent space, where all the shapes are concentrated in the middle, and it's mostly triangles. So we have this sort of the selection process that is sort of revealing this it's sort of novelty first. You saw before where it was really clustered in the corner of the latent space, but this is where we have, I think, a good representation of different shapes and different variation. And then, but what we can do with this, though, is we can actually create a phylogeny. So remember, we have this latent space that we sample from and we select from, and then the outputs of selection are in this sort of in this space of, or in this language of different shapes. And so, but what we can do actually is create a dendrogram, which is a form of tree. And we can plot this out basically where we have this novelty first dendrogram. So this is where we have this tree, and we have some clusters, and we have some branches, some lineages in here. And basically what we're doing is we're doing hierarchical clustering based on prior shape history. So we're encouraging novelty, and we're using that prior shape history to form the tree itself. And so we can see here that in the paper I talk about some of the interesting features of this tree. There is a problem with interpretability here because, again, we're making this, we're using the types of projections that we have available to us. Ideally, we'd like to have a very specific method for forming these phylogenies, maybe like neighbor joining. We could use something like cladistics, but this is all kind of, for later work. The next data set I'd like to talk about is the Bradenburg vehicles data set. So this is where we go back to Valentino Bradenburg's book Vehicles, and we mine that book. We train the model in the book so that it knows that there are different vehicle phenotypes, there are different sub-vehicle phenotypes. So this is vehicle 1, vehicle 2, and vehicle 8 is defined by Breitenberg. And what we want to do is we want to break those phenotypes into parts. We want to sample and generate morphologies of vehicle bodies. And then we want to sample and generate descriptions of behaviors and associated connectivity patterns. So as you can see, there are connectivity patterns between sensor and effector, and all of those things produce what Breitenberg described as different behaviors, which, there are things like tropotaxis and chemotaxis and spatial behaviors that are sort of, they look like those things in nature. And then he went further and described those things as like fear and love and other things, which is a problem with maybe the source data. But what we're doing is we're going to use that to form our phylogenies and to form our sort of the way in which these data are put together. So again, if we go back to our work that we've presented at the Embodied Intelligence Workshop in 2025, we presented this work on phylogenetic models of embodied agents, an eco-evo-devo approach. So in this work, we actually evolved or showed phylogenies of Breitenberg vehicles, with vehicle 1A being the common ancestor, and then evolving into two lineages, or two clades, one of vehicle 2 and one of vehicle 3. And so then that's kind of our criterion for, or our starting point here. And what we show on the right-hand side of the slide is that we have vehicle 2A, vehicle 3A, vehicle 3B, and then we have all of these sort of endpoints of this phylogeny phylogenetically mixed into this new vehicle here. So this vehicle up here, this phylogenetically mixed vehicle, is a combination of these three architectures. This increases the complexity of our circuit, and it does so from common ancestry. In this case, what we're showing is a hypergraph, which is a bipartite graph, and it shows different concepts or different components, and it links it with different vehicles. So in this case, we have things like excitatory, crossed, complexity, rules, spatial, and it just linked to different vehicles. So we kind of know what we're getting when we take different vehicles and we cross them, or we do this in silico hybridogenesis. So if we combine vehicle 1 and vehicle 10 and vehicle 11, we have different associated behaviors. So we derive the morphologies and behaviors from Valentino Breitenberg's book Vehicles, Experiments in Synthetic Psychology. We reconstruct the embodied nature of a sensor effector mapping. So that's what we're trying to do here. Instead of using just shapes where it's kind of abstract, we're actually building very simple versions of these embodied agents. This is an example of what we're doing here. This is an example of the output of the reservoir network. Sometimes they call reservoir networks echo state networks, so that just a point on terminology there. But what we've done here is, in our reservoir example, we've generated 50 vehicle phenotypes, or 50 vehicle configurations. And so we can see that these circles embody the sensors, effectors, and the inhibitory and excitatory connections between them. So this is the description of these vehicles, the variation that we're producing. We can also do this with forward and reverse diffusion, and this on the bottom is the same sort of output of 50 vehicles, but we do this with this method of forward and reverse diffusion. So not a lot of difference between them. In fact, they almost look exactly the same. So there isn't a huge amount, at least for this data set, of difference between these two methods. Now, as I said before, after post phylogeny construction, we want to then take those components and assemble them into these hypergraphs. So then we ask the question, what are hypergraphs? And so hypergraphs are these networks that have hypernodes and hyperedges. And what we do in the hypernodes is we classify different nodes and edges from like a regular network, and we just have the second level of organization. So for each hypernode, which are these large circles, we might have 5 or 6 nodes within them that are all connected internally. And then this other hypernode might have 5 or 6 nodes connected internally. And then there are connections that cross the boundaries of these hypernodes. those connections across the boundary are the hyper edges, which is just a bundle of all of these connections between the two hypernodes. So we might take, say, for example, a hypernode that represents one type of vehicle, a hypernode that represents another type of vehicle, and they're tied together by these relationships between them. So in this case, we have a hypergraph, and this is sort of an abstract version of this where we're building this architecture out. The hyper edges connect different parts of the architecture together. So the hyper edges are critical in connecting the two types of things together. And so this becomes an issue in some of these wildly different components. So our architecture includes components that have undergone evolution in different lineages, and we're bringing them together, and we have to be mindful of how they're coming together and if they're compatible. Hypernodes, of course, diversify the architecture and provide the basis for complex circuits. So our hypernodes are the components of our architecture. So like in the Bradenburg vehicles example, we had, say, vehicle 1, vehicle 3, vehicle 8. And those would form, each of those vehicle architectures would form a hypernode that would then be combined into a hypergraph of representation of that mixed architecture. And so this is actually from some of the Bradenburg vehicle work, so it's a little bit more concrete. This is an evolutionary process of 5 steps, so it's a very simple evolutionary process where we have these different vehicle initial states. We have vehicle 0, 1, 2, and 3, and then we evolve each of those over time. We form these lineages, so we see these arrows. We evolve vehicle 1, and we generate a vehicle 0 in generation 1 that's similar but different. And we have these lineages that maybe have dead ends and don't branch, or we have lineages that branch and kind of form this internal phylogeny. And what we're going to do here is we're going to take this entire structure, this entire sort of, it's not really a phylogenetic tree in the conventional sense, because it has multiple common ancestors. But what we're going to do is we're going to sample these phenotypes or these vehicle types, and we're going to put them into a hypergraph. So this is a phylogenetic model of Bradenburg vehicles. We have various emerging lineages of vehicle morphology. Some proliferate. So, for example, they branch into 2 different lineages or 2 different sublineages. Sometimes they're dead ends. But whatever happens, we want to treat each one of these examples as a potential component for our hypergraph. So what we're looking at here is evolution in fits and starts. As Stephen Jay Gould used to say, we have our products, which could be endpoints of a phylogeny, or transitional forms, which could be in the middle of a lineage. And these all contribute to hypergraph architectures. So again, we have these hypergraphs. And one of the things about hypergraphs is it not only gives us this sort of higher meta level of organization, but it allows us to have these multi-scale relations, which are very important in representing complex systems and biologically inspired systems. So this is an example of our hypergraph with connected hypernodes. So again, we're taking these nodes from our Bradenburg vehicle phylogeny, and we're connecting them together. And then we have this hyper edge that connects these 2 components together. And so that's what we have here. We have this description listed down here. So, we have this hypernode on the left, which is from generation 4 vehicle 3 plus generation 2 vehicle 0. That's one component. And then that's linked to another component, which contains generation for vehicle 0 plus generation for vehicle 1 plus generation for vehicle 2. And that's another component. And so that all works together as one of these phylogenetically mixed architectures. So one of the final things I want to talk about here is how do we make this pipeline more ecologically friendly? We want to be able to, we used Copilot to build the initial pipeline, but that does take a lot of resources. It generates a lot of carbon emissions. And so we want to be able to lower carbon emissions for the pipeline. So we're mindful of that. So especially when you scale this up. So, one thing we considered was, will running locally with open source models rather than frontier models help? And it may help. So we can look at different open source models and see which ones, especially if we run them, say, like on a Mac Mini, we can actually do this a little bit more easily. The second point is, what if we use RAGs more strategically to precisely define the training set? So like what we did with the Bratenberg vehicle example, but we really kind of refine our training set, and we really have something that is extremely refined. So I talked about before how we can analyze different small connectomes. If we could use small connectomes like C. elegans or Drosophila, which isn't as small as C. elegans, but it's something that we can manage computationally, could we use those to sort of create biologically inspired components for an agent? Sure, we could probably do that. And, this is important when we scale things up. So we, especially when our data set is not just shapes, when it's something like our Bratenberg vehicles example, and sometimes we have a training set that's in 3 dimensions or more higher dimensions. And so that's going to be especially important to really refine the training set and really refine how the model is responding to the nuances that you want to capture. And then finally, we have these strategies for carbon prompting. So there's this term in the literature called carbon prompting, which is where you try to prompt your model in a way that's mindful of carbon emissions. And so this is where the strategy is to use as few prompts as possible to generate a sparse representation. Once you refine the pipeline, then you minimize the number of prompts used. So you start off by kind of figuring out what your prompt should be. You go through and you look at what the problems are, and then you really refine your pipeline, and you try not to use too many prompts. So you try to kind of infer how to best refine your pipeline, or to really kind of get your hands around your pipeline and how it works, but not prompt it every time you have a question. So there are a lot of strategies we can pursue for this. So thank you for your attention. We have our code and other materials in our GitHub repository, which is here. This is at github.com, Bradley Alicea, Phylogenetically-mixed Architectures. We also have another presentation at Artificial Life 2026, Toy Models of Early Life. It's going to be presented at LifeDef 2026. But there's a video of this presentation on the DevoWorm YouTube channel. So look for that in the DevoWorm YouTube channel. This work has been done in conjunction with the Computational Developmental Systems Interest Group and the Representational Brains and Phenotypes Interest Group at the Orthogonal Research and Education Lab.